Hello, you're watching a Unipro Eugene podcast. Here we discuss the hottest topics introduced by our users and show some helpful tips for using Eugene, a free cross-platform genome analysis suite. In this episode, we're going to show you new features that appeared in UGENE 1.12. A possibility to search for subsequences using several patterns was added to the sequence view. Use Ctrl plus Enter keys to input several patterns. You can either input the patterns line by line, or you can input them into the FASTA format. Another option is to load patterns from a file. The file can be in any sequence format supported by Eugene. Note that when the input patterns are in FASTA or other sequence format, the names of the pattern sequences can be used as the names of the annotations found. Check the Use Pattern Name checkbox to enable this option. Now let's find two patterns in this sequence. You can see that the names of the annotations are the same as the names of the input sequences. The panel with options appeared in the alignment editor. On the highlighting tab of the panel, you can select a color scheme of a multiple alignment. For example, let's select Jaw View instead of the default Eugene color scheme. There are different modes of highlighting of alignments. Most of them require a reference sequence to be selected. Let's select, for example, the first sequence as a reference. Right-click on the sequence and select the corresponding item in the context menu. Let's select Agreements highlighting mode. This means that if a character at a position in a non-reference sequence is the same as a character at the same position in the reference sequence, it must be highlighted. We can also check Use Dots to replace all non-highlighted characters with dots. Pairwise alignment of two sequences is now available in the alignment editor. You can select either Hirschberg or Smith-Waterman algorithm to do the alignment. Select the sequences. If required, configure the algorithm settings. The result of the pairwise alignment can either be shown in the current multiple alignment window or opened in a new window. Let's show it in the current window. One more major option that appeared in the alignment editor is the possibility to add a tree, or even several trees, synchronized with the opened alignment. Let's add a tree. First of all, we're going to sort the sequences of the alignment in the same order as the sequences in the tree. To do that, press the corresponding button on the toolbar. You can modify the tree appearance, for example, by configuring the tree settings on the Options panel. For example, let's modify the tree color and the width of the tree lines. Once the tree is added in the alignment editor, you can easily see clusters of sequences. Move the clusterization line of the tree. Based on the cutoff you specify, the sequences of different clusters are highlighted with different colors. The way you specify input data in the data reader's workflow elements in the Workflow Designer has been modified. Now you can put your input data into different datasets that would be analyzed separately. Each dataset may consist of one or several files. You can also specify one or several directories to read the input files from. Additional settings are available to filter the input data. For example, let's select a GenBank file and a directory with FASTA files as input for our workflow. Several workflows for analyzing next-generation sequencing data appeared in UGENE 1.12. The pipelines are available on the Samples tab of the Workflow Designer palette. There are call variants with SAMTools pipeline, Systrome pipeline for ChIP-seq analysis, and Tuxedo pipelines for single or paired input RNA-seq short reads. The Tuxedo pipelines are available on Linux and Mac OS systems in the current Eugene version. Let's select the first pipeline. To set up the scheme parameters, we're going to use a wizard. Note that such wizards are currently available only for the NGS samples. On the first page of the wizard, we need to input a reference sequence and an assembly file in BAM or SAM format. The second and the third pages allow configuring SAMTools parameters. We will leave the default values. Finally, on the fourth page, we need to specify an output file with variations. Now the scheme is ready to be executed. A simpler but not yet so effective and precise way to search for variations is compare consensus and reference sequences.
To export such variations for an assembly opened in the assembly browser, select Export Consensus Variation item in the context menu. In the Appeared dialog, you may select Simple SNP or VCF4 variations file format and configure other parameters. A special Eugene extension for web browsers was developed that allows one to easily load data from the web into Eugene. The extension is currently available for Google Chrome and Mozilla Firefox web browsers. To try out the extension, add it to your web browser and launch UGENE 1.12. Now let's go to the BioMart portal. This system enables a scientist to perform advanced querying of a wide range of biological data sources through a single web interface. Let's proceed to BioPortal. The current version of Eugene Web Extension can detect the following types of identifiers: Ensemble Gene ID, Ensemble Protein ID, and PDB ID. You can see that the background color of the detected ID has been changed to a light blue color. Right-click on the ID. Open in Eugene item is now present in the context menu. Click on the item. Now, open the Eugene window. A sequence with the specified identifier has been loaded into Eugene. The extension also allows you to open data selected on a web page in Eugene. Refer to the Eugene documentation for details. These are only some of the major features that appeared in Eugene 1.12. Visit our website to learn more about Eugene and other features that appeared in version 1.12. Feel free to write us your questions and ideas about Eugene, and stay tuned for new episodes.